All right, so welcome to Concept 2 Notes and Unit 4 Genetics for Protein Synthesis. So we just finished, we just wrapped up DNA structure and replication, and now we're going to talk about something a little bit different. So DNA replication has more to do with mitosis and the cell cycle than it has to do with making protein. So everything we've been talking about, DNA and our genes and all that. Replication is more, like I just said, is more about the cell cycle. However, the only reason we teach these at the same time, replication and protein synthesis, is because they are a very similar process. They use some of the same kind of mechanisms, and the whole point is just that we don't want to teach about base pairs twice. We don't want to teach you about the A's and the T's and the C's and the G's before it's absolutely necessary, and so that's why that's why replication is kind of included in this unit.
After learning about DNA, have you ever wondered how can the DNA actually result in a trait? Let's take an example, like eye color. Yes, your DNA has the genetic information that codes for the color of your eyes. Your eye color is based on a pigment that is inside the eyes, but in order to have that pigment, you have genes, which are portions of DNA that can code for proteins which help make that pigment. So what we're going to talk about is how your DNA can lead to the making of a protein. This process is called protein synthesis. Synthesis essentially means to make something, so protein synthesis means to make protein. And you may wonder, what's a big deal about proteins? Well, you may not realize this, but proteins are kind of a big deal. They do all kinds of things. Proteins are involved in transport, in structure, in acting as enzymes that make all kinds of materials, in protecting the body, and so much more. You've got to make proteins. It's essential for you to live. And what is so cool is that you are making proteins right now as you sit and watch this video. It's happening in your cells. They're making proteins. So back to your DNA and its role in all of this. All of your cells have DNA. Well, a few exceptions. And that DNA is in the nucleus. Some DNA is non-coding DNA. Some DNA makes up genes that are not activated. More about that in our gene regulation video, but we're going to talk about genes that are coding for active proteins. So how are we going to get the information from these genes out of the nucleus so that the cell can start producing the proteins that it needs to make?
All right, so protein synthesis. Protein synthesis just means it is the process of reading the instructions in our DNA to make a protein. Okay, so there is this big idea in genetics, and it's called the central dogma of genetics. And don't get too frightened by the wording here. It just basically is this big overarching idea that everybody kind of accepts and everyone kind of knows about. So this idea, the central dogma states that DNA, like I just said, are instructions, and they are located within the nucleus and they cannot leave. And like we've mentioned time and time again before, it's a good thing that our DNA doesn't leave, because when it's inside the nucleus, it's nice and protected. And if it were to be able to leave, things might get a little hairy. However, this is a problem, because if you'll recall from our cell organelles unit, proteins are made by ribosomes. Remember those things, those green things that are found in the cytoplasm, and they're found like attached to the rough ER that's like around the nucleus? Yeah, that thing. So proteins are made in the ribosomes outside, and so to rectify this. To fix this problem, solve this problem, our cells fix it in 2 steps. So this is, again, the central dogma of genetics states all of these things. The central dogma states that our DNA gets converted into RNA, or is transcribed into RNA, and is copied into RNA. And then that RNA template is what leads to the formation of a protein. And so step number 1 is called transcription. And so that's just, we're going to talk a little bit more about that in a second, but DNA is being read, is being copied into a form that can leave the nucleus. And then that copy is then translated into forming a protein. Okay, remember proteins? So proteins, in case you forgot, are the most versatile macromolecules in living systems, and they serve crucial functions in pretty much all biological processes. Like they function as catalysts, so enzymes. They speed up chemical reactions by lowering activation energy. They transport and store other molecules like oxygen, their hormones. They provide mechanical support and protection for our immune system. They generate movement, so proteins, help build our muscles. And they also help with transmitting nerve impulses. So with sending a signal from your brain, say to your finger to your hand, so like maybe go like that. You're going to give someone a high five, that you are transmitting a nerve impulse from your brain saying, hey, hey, hand, raise, and high five that person. So proteins have a role in that. And then they also have a role in controlling growth. Some are purposes of mitosis, growth and repair. So they control growth, and also cell differentiation. Remember, when cells get their jobs, they become either a cardiac cell or a bone cell, skin cell, yada yada. Hashtag proteins matter. Don't forget it. Remember it. Get it trending on Twitter. But although apparently you guys tell me Twitter is not a thing anymore, yeah, okay, whatever. Get it trending somewhere. Okay, so we're gonna go back to RNA for a second, and I know we talked a little bit about it in the last video, but so nucleic acids are a type of macromolecule, right? There are two types of macromolecules that we're talking about. Well, there are only two in existence: DNA and RNA. Okay, DNA, deoxyribonucleic acid, and RNA, ribonucleic acid. So within RNA, there are three different types of RNA that it can be. The first is called mRNA, or messenger RNA. And just kind of write down these definitions for now, and we'll get in. You'll see why it'll be necessary to have these definitions in a second. So our mRNA. Messenger RNA copies instructions in our DNA and carries these to the ribosomes in the cytoplasm. So here is a picture of what mRNA looks like, and so it looks exactly like what you would think. This is kind of the one that this is what you guys have already kind of learned about when I taught you about RNA. So yeah, just that single-strand molecule ribose sugar. Okay, the second type of RNA is called transfer RNA, or tRNA, and this type of RNA carries amino acids to the ribosome and the mRNA. So right here is our tRNA molecule. We got transfer RNA, and it has 3 nucleotides attached to it, 2 nucleotides of RNA, and then over here it is carrying an amino acid, and so this is going to be important when we actually get to the ribosome and begin step 2, which is translation. And this next part is just basically, I'm just telling you that this exists. There is a third type of RNA called rRNA, or ribosomal RNA, and just know it composes the ribosome. It is a molecule that forms the ribosome, and that's all you need to know about that. Okay, so let's actually talk about protein synthesis now that we got all that out of the way. So step one of protein synthesis is transcription. And so I'm going to walk you through a little story really quickly before we get into that. Let's say that your grandmother is an incredible baker. She makes the best brownies, she makes the best cookies, and she has tons and tons and tons of her own original recipes in a cookbook that she keeps in her kitchen all the time. And let's say one day you go over to grandma's house and you ask her for her amazing brownie recipe. Now, her recipe book, mind you, is very large. There are like 200 recipes in that thing. It is super heavy, and Grandma, she doesn't want you to lose it. She'd be afraid. She would not want you to take that entire recipe book home. I mean, number one, what if you lost it? I mean, then she'd be out all of those recipes. She would lose all of that. And number two, it just doesn't really make that much sense. If you only need that one recipe, you only want that one copy, it doesn't make sense to take the whole book, right? Right. So what would you do to solve this problem? And I think most of you would probably say, oh, I would just take a picture of it with my phone. Perfect. Or you could take out a piece of paper and copy the recipe down on a physical piece of paper. You could take it to a copy machine and just kind of place it in the copy machine and then print out and then have a printout of it. All very good options. And so this scenario is very similar to what happens in our own cells. DNA can never leave the nucleus. And no one's complaining about this because our DNA is very, very, very important, and if something would ever happen to it, it's very damaging. It could lead to death. So we have to have a way of getting that DNA, those instructions for building those proteins, out of the nucleus and into the cytoplasm, where it can be delivered to the ribosome. Because remember, the ribosome is what actually makes proteins. They're little protein-making factories. So the purpose of transcription is to carry the code or instructions out of the nucleus. Yep, so I just said all of that. Perfect. So the location of transcription is within the nucleus. And we say that it starts with DNA. So at the beginning of transcription, you start with DNA. And at the end of transcription, you end with mRNA. So remember that messenger RNA. And that will be the copy that we will use to begin stage 2. Okay, so I know that probably didn't make very much sense. It's okay. Let's actually talk about what is happening on the cellular level. So the process. First, we have to unzip the gene that needs to be copied, right? Because we're trying to build a protein, and we need those instructions. And genes are very specific sections of DNA that contain those instructions. So we unzip it, just like we did in DNA replication. This time, an enzyme called RNA polymerase uses complementary base pairing rules to match RNA nucleotides with the exposed DNA nucleotides, very similar to what we did in replication. However, remember, RNA has uracil in place of thymine, so A binds with U, not T. So like in DNA replication, when we had when we had DNA polymerase adding complementary nucleotides to this new strand, and all of that was happening on both sides in replication. When we're talking about protein synthesis, transcription, we only need one of the we only need one strand. We only need one half of these instructions. We don't need to do anything with this other strand, that other half of the DNA molecule. We're only looking at just one side. So this bottom side is one that we are focusing on. So RNA polymerase, pretend RNA polymerase is kind of like right here, and he is going down along here, and he has added nucleotides, like we just said, using those complementary base pair rules. So remember, we're using U, not T. So he binds U with A, C with G, A with T, U and A, GC, U and A, and so on and so forth. And he is making a copy of this section of that DNA. And you can see that it's continuing off over here because you can see here the DNA is being zipped back up as soon as it's done. Okay. So after RNA polymerase adds those complementary nucleotides, the completed mRNA molecule is released. Remember, mRNA means messenger RNA. So think of it as kind of like a mailman delivering the instructions. So he copies down the instructions and then he delivers it to where it needs to go. So this strand right here will get released, and the DNA will zip back up, and the mRNA will leave the nucleus and enter the cytoplasm. So here is just another visual representation. I'm going to show you another one in just a second. So transcription, DNA is copied into a complementary strand of mRNA. If you'll remember, I had you guys predict how you thought we managed to get those instructions out of the nucleus, and you guys had some predictions, and so this is it. This is the answer. We copy it into mRNA, and so here you can see our double strands of DNA, and then here you can see we have unzipped. So here is our left side strand of DNA, and then on this side we have the right side of the strand, and then here we have created a complementary copy of this gene in mRNA. So here is the mRNA, and you can tell it's mRNA because you can see the uracil there in place of thymine. Okay, and then after we're done, that mRNA, so this guy right here, will then be released, and the DNA molecule will zip itself back up. So you end up with one completed DNA strand, so it goes right back to what it looked like before. And now we have this mRNA copy that contains those instructions, contains the sequence of nucleotides that will allow us to code for that gene, that protein. Okay. So why don't you try it? Transcription. I want you to try transcribing a sequence. So if I gave you this DNA sequence, TAC, GCT, AGT, ACG, ATT. I want you to try and guess, well, not guess, I want you to put together a complementary sequence, a complementary strand of mRNA. So what you would do, I'm going to do the first three for you. So if we had T, your complementary mRNA nucleotide would be A adenine. For A, your complementary mRNA nucleotide would be U. U, because remember, A does not pair with T. We're talking about RNA, DNA, RNA, DNA. So first we already have A, then we have U, and then C is going to pair with G. All right, so our first three nucleotides in our mRNA sequence are A, U, G. Now I want you to finish putting together this sequence. Okay, ready, go. Okay, so drum roll please. If you said AUG, CGA, UCA, UGC, UAA, you're correct. Nice job. You can go ahead and practice this on your own. You can come up with your own sequences in their own order and test yourself and get really good at this and really think about the difference between DNA and RNA. We're just copying the DNA on that one side, the RNA is on the right. Okay, so we're going to see T's on the left, but we're not going to see T's on the right. We're going to see U's. Okay, all right. That concludes the transcription section of protein synthesis. And all right, enjoy.